Renal tubular processing across the distal convoluted tubule and the collecting tubule. We will be looking into the importance of different transporters as well as the clinical correlations of transport across these different tubular segments. So starting from the early distal convoluted tubule, as you can appreciate in this image, the importance of the macula densa that we have discussed in our session on the tubulo-glomerular feedback as well. The special role in the dilution of urine we will be discussing in a coming up session. This early distal convoluted tubule absorbs approximately 5% of the filtered load of sodium, potassium, and chloride by this sodium chloride co-transporter. You can appreciate again the sodium potassium ATPase pump on the basolateral membrane. Once the chloride is being absorbed, it passively diffuses by the leaky channels into the interstitium. This segment is again impermeable to water and urea like that of the thick ascending limb of loop of Henle. That is why we also call it the diluting segment, as the salts are being reabsorbed continuously but water is left behind, thus diluting the tubular fluid content further. The thiazide diuretics can inhibit the sodium chloride channels, thus resulting in excretion of sodium, calcium chloride as well as the potassium in the urine. Processing across the late distal tubule and cortical collecting tubules. They approximately behave similar to each other. Their tubular membrane is having two types of the cells, principal cells and intercalated cells, which further are of two types, type A and type B cells, but principal cells are more in number as compared to the intercalated cells. Here you can have the comparative image of the role of early distal convoluted tubule in the absorption of sodium chloride, calcium, magnesium, whereas the role of principal cells in the reabsorption of sodium and chloride, secretion of potassium. Reabsorption of water is also occurring over here in the presence of ADH, although the early distal convoluted tubule was virtually impermeable. Type A intercalated cells are involved in the reabsorption of potassium and bicarbonate and secretion of the hydrogen ions. We will be looking into the special roles of these cells in the maintenance of urine osmolarity as well as pH in the coming up sessions. So starting from the principal cells, you can appreciate the sodium potassium ATPase pump on the basolateral membrane, which is creating a concentration gradient for the sodium between the tubular lumen and the cell, which is going to lead to the absorption of the sodium across these channels and the release of the potassium into the tubular lumen simply by applying the concentration gradient rule. Also, it has the passive leaky channels for the chloride reabsorption. Here, this is also a potential therapeutic target for the action of aldosterone antagonist and sodium channel blockers, because aldosterone over here not only enhances the sodium potassium pumps but also translocates more sodium channels on the luminal membrane for the increased reabsorption of sodium. In comparison, the intercalated cells type A, although still involved in the reabsorption of potassium, are also involved in the secretion of the hydrogen ions. At the level of intercalated cells, potassium and hydrogen ions compete with each other. If we want to excrete more hydrogen ions, type A cells become more active. If we want to excrete more potassium ions, type B cells become more active. They act in exact mirror image of each other. As you can see the active secretion of hydrogen ions by the primary active transport as well as primary active hydrogen potassium pump over here, and the reabsorption of potassium by these potassium leaky channels, reabsorption of bicarbonate by the chloride bicarbonate channels at the level of the type A intercalated cells. This is occurring by the carbonic anhydrase mechanism, whereas the type B cells are exactly opposite of this. They lead to the secretion of the potassium by the concentration gradient and reabsorption of the hydrogen by these primary active transporters, hydrogen ATPase and hydrogen potassium ATPase. Bicarbonate ions that were being reabsorbed by the type A intercalated cells are now being excreted by the chloride bicarbonate exchanger at the level of type B intercalated cells. So this is the body potassium and acid-base balance status, which decides if type A cells are going to be more active or type B. So if there is chronic acidosis and we want to get rid of hydrogen ions, of course, type A cells are going to be more active as compared to the type B cells. Late distal and cortical collecting tubules are the potential site of target of aldosterone, as they can increase the insertion of epithelial sodium channels and sodium potassium ATPase, and also they are the potential site of target for aldosterone antagonist and sodium channel blockers, which are the very powerful diuretics. Late distal and cortical connecting tubules secrete hydrogen ions by primary active transport, as you have seen, by hydrogen ATPase and hydrogen potassium ATPase, whereas in proximal convoluted tubule, this was being secreted by the sodium hydrogen counter transport. So they have a special role to play in the acid-base regulation as well. These are completely impermeable to urea segments. Water reabsorption is subjected to the presence of antidiuretic hormone. In the absence of ADH, they may become completely impermeable to water. They are also completely impermeable to urea, especially the cortical collecting tubular segment. The solute reabsorption here is regulated by aldosterone and tubular flow rate. Secretion of hydrogen ions is occurring by the primary active transport mechanism, and water reabsorption is subjected to the availability of ADH. This is what is the summary of reabsorption across the late distal tubule and cortical collecting tubule. In comparison, if we look into the medullary collecting duct, this is having again cuboidal epithelial cells, pure mitochondria, smoother surface, and is having principal and intercalated cells again. It is involved in reabsorption of 5% of the filtered load, which is again modulated by the vasopressin and aldosterone. They have again a special role to play in final composition of the urine and deciding its osmolarity. If we compare the cortical collecting duct with the medullary collecting duct, both are permeable to water only in the presence of ADH. The cortical collecting duct is impermeable to urea, but medullary collecting duct becomes permeable, especially under the presence of ADH, and both play an important role in acid-base balance by secreting the hydrogen by the primary active transport mechanisms. And this image is showing the tubular fluid to glomerular fluid concentration, increasing for the substances which are secreted actively and decreasing for the substances which are reabsorbed. Especially at the level of distal and collecting tubule, you can see increased level of potassium. And this is summarizing what we have just discussed: early distal convoluted tubule, late distal convoluted tubule, cortical and medullary collecting tubule. And is a typographical error. This early distal tubule is impermeable to water. Rest we have discussed previously. As these segments are impermeable to water in the absence of ADH, the fluid content over here is hypoosmotic, which may remain hypoosmotic or become hyperosmotic depending upon the availability of the ADH. Sodium handling across the nephron, 67% is being reabsorbed at the proximal convoluted tubule level, 25% at the thick ascending limb, 5% in the distal convoluted tubule, and this 3% is subjected to variation in the collecting tubules depending upon the presence or absence of the aldosterone. This is summarizing the sodium chloride transport across the different segments of the tubule along with their regulation. And this is summarizing the water transport across the different segments of the tubule that we will be reviewing again while discussing the urine concentration and dilution mechanisms. Thank you very much.